guys, good evening once again and welcome to my YouTube channel. Today we'll be discussing about the structure of the prokaryotic cell or the bacterial cell. And we are going to talk about the various parts of the bacterial cell and then their function. So first of all, we'll start by talking about the cell envelope and appendages. Then secondly, we'll talk about the cell interior or the cellular interior. Now, we are pretty much aware that there are basically two types of cells. We have the eukaryotic cell and the prokaryotic cell. So what we know of the eukaryotic cell is that they have membrane bound structures, and for these ones their nucleus are not scattered in their cytoplasm. The eukaryotic cell, their nucleus are not scattered in their cytoplasm. But for the prokaryotic cell, we can talk about it being a non-membrane bound. So they don't have membrane bound structures, and then their nuclear materials are scattered in their cytoplasm. So nuclear materials are scattered in their cytoplasm. So that is one feature of the prokaryotic cell. So the diagram we have here is the structure of a simple prokaryotic cell, the bacteria. So unlike other eukaryotes, the prokaryotic cell is different at all. It's not like the eukaryotic cell. So the prokaryotic cell mostly is unicellular, whilst the eukaryotic can be multicellular. So the prokaryotic cell, for instance, it can exist on its own, unlike the eukaryotic that has to get two or more in order for it to survive. So this one is unicellular. So we can see that we have the various parts like the DNA coiled into nuclei. So this is the nuclear materials. It is not enclosed in the nucleus, but it is scattered in the cytoplasm. Then they have ribosomes, they have cytoplasms, and they have cell wall and cell membrane. So this is their cell wall, then we have their cell membrane or plasma membrane. So unlike eukaryotic cells that do not have cell wall, prokaryotic cells have cell wall. But eukaryotic cells do not have cell wall. So we realize that the prokaryotic cell is somehow distinct or different from the eukaryotic cell. And today we are going to talk about each of the components and go much deeper into it. So now we have our structure. We have our cell wall. So the bacterial cell has a cell wall that encase it. Then it has ribosome just like any other ordinary cells. Then we have the cytoplasm. Then we have the cell membrane. So before you can get into the cell, first of all you have to destroy the cell wall. Then you have to get to the cell membrane. Then we have the flagellum. So flagellum is basically for movement, and it is part of the cell appendages. It's part of the cell appendages. Then we have the pili. So in bacteria, this pili is also for attachment. This pili is for attachment. Okay, not necessarily for movement. The main structure that is for movement is the flagellum, but the pili is actually for attachment. And in some bacteria for sex, we have sexual pili as well. So they involve the pili in sexual activities. So we have sexual pili and then the one that we use for attachment. So these ones that you see here is the pili. Okay. So this is actually the basic structure that we can briefly talk about. Now let's go a little much deeper into it. So we have cell envelope and appendages. So the cell envelope and appendages, we have one. Then make this I, the cell wall. The cell wall. Then we have the cell wall, we have the cell membrane, or the plasma membrane. Then under the cell appendages, we have the flagellum, we have the pili, we have the capsule, and then we have the fimbri. So that will be it. So now let's start with the cell wall. So the cell wall of a bacteria. Now the bacterial cell wall is a very complex cell wall. It's a very complex cell wall. So what we have to notice about the bacterial cell wall is that the bacterial cell wall is made up of a peptidoglycan layer. Peptidoglycan layer. So let me just give a sketch of how the bacterial cell wall is. So the peptidoglycan layer actually itself is made up of two molecules. So we have N-acetyl glucosamine and N-acetyl murin or muramic acid. N-acetyl muramic acid. And these are cross-linked by peptide bond. So this is how the cell wall of the bacteria looks like. So this is how the cell wall of the bacteria looks like. So let's say this one to be N-acetyl glucosamine. Then we have this one to be N-acetyl murin. N-acetyl murin. Or muramic acid. You can say N-acetylmurin or N-acetylmuramic acid. So they are the same. And these ones are what? They are cross links by peptide bonds. So peptide bonds. They are cross linked by peptide bonds. So this is basically the structure of the what? Of the cell wall. Now, this structure of the cell wall has distinguished the bacteria into two categories. So by virtue of the cell wall, bacteria has been classified into gram positive bacteria, and then we have gram negative bacteria. So these are the two types of bacteria that we have according to what? According to the cell wall. So what we can say about the gram positive bacteria is that for the gram positive bacteria, so they have thick peptidoglycan layer. And then the gram negative bacteria have what? So they have thin peptidoglycan layer. So the cell wall distinguish the bacteria into two forms: gram positive bacteria and gram negative bacteria. Gram positive bacteria have thick peptidoglycan layer, and gram negative bacteria have thin peptidoglycan layer. Now, the gram positive bacteria cell wall structure is somehow different or has different chemical constituents than the gram negative bacteria. So let's talk about the components of the gram-positive cell wall, the major components of the gram-positive cell wall. Okay. So for the gram-positive cell wall, these are some of the chemical constituents that we have. So one, so gram-positive cell wall. So these are some of the constituents we have. So we have teichoic acid, then we have lipoteichoic acid, then we have peptidoglycan layer. So that is for the gram-positive cell wall. We have teichoic acid, we have lipoteichoic acid, and then we have peptidoglycan layer. So for the gram-negative cell wall, we have lipopolysaccharide, we have lipoproteins, we have purines, or what we call the porins, then we have phospholipids, and of course the peptidoglycan layer. Now if you look at these two bacteria, you realize that the gram-negative cell wall is a little bit complex than the gram-positive. For the gram-positive, we only have teichoic acid, lipoteichoic acid, and the peptidoglycan layer. But for the gram-negative, we have lipopolysaccharide, lipoproteins, purines, phospholipids, and peptidoglycan layer. So this one is more complex than this one. Now let us go through how we will be able to distinguish between the gram-positive bacteria and the gram-negative bacteria. So there is a procedure we call gram staining. So in this is a staining approach in which we stain the bacteria to identify it under a microscope, whether it is a gram-positive bacteria or the gram-negative bacteria. So in this gram staining, I'm going to take you through the practical process, then we will explain theoretically why after the staining some bacteria will tend to be gram-positive and others will tend to be gram-negative. So now in this process, we have stains that we use. So first of all, we have the crystal violet. We have crystal violets. Now we have the iodine solution. Then we have our alcohol or decolorization agent. Then we have our counter stain. In some other books you might see safranin. That is our counter stain. So this is the reagents that we use. Okay. So first of all, what we want to do is that we will prepare a smear on a slide. So this is what is going to be. This is our slide. We'll make sure that we prepare a smear on our slide. So then we allow the smear to dry. Then after that, we pour crystal violet onto the smear to stain the smear. So this one is the primary stain. So we apply crystal violet. So first of all, we make a smear. Then secondly, we apply what? The crystal violet solution. Then what we are going to do is that we are going to wash the slides, and after that we pour our iodine solution. So we pour our iodine solution here. So here we are going to get our iodine solution. We pour our iodine solution, and then we are going to wash. After that, what we will do is that we will use our decolorization agent, that is the alcohol. So here we will use alcohol to wash and decolorize. Then finally we use our counter stain, that is the saffron counter stain. So the first stain, crystal violet, what it does is that it impacts color onto the cell wall of the bacteria. And what the mordant does, that's the iodine. The iodine solution is also called mordant. What the mordant does is that it forms a complex with the crystal violet. So it becomes a crystal violet iodine complex. And this complex is much absorbable, or is much retained by the cell wall of the bacteria. So that is the main reason for the iodine solution. It's a mordant, it forms a complex with the crystal violet, and that complex is readily absorbed or taken by the cells of the bacteria, or by the bacteria cell wall. And then what the alcohol does, the alcohol is a decolorization agent. What it does is that it decolorizes the new crystal violet iodine complex that has formed. It takes off the iodine solution from the cells. It takes off that color. And now finally the counter stain. The counter stain is actually to detect what? Gram-negative bacteria. Alright, so we'll explain. So this is the practical aspect, what is going to be done at the lab. Now the theoretical aspect is that for gram-positive bacteria, they have fake. So what we have to take notice. So gram-positive have fake. So they have fake peptidoglycan layer. So they have fake peptidoglycan layer. So as a result, during the decolorization process, due to the fake peptidoglycan layer, the alcohol will not be able to, take off the iodine solution. So even after counter stain, or after the use of the safranin, the safranin is pink in color. The safranin will not be able, the cells will not be able to absorb the safranin, because the decolorization process did not take away enough enough crystal violet. They didn't take away enough crystal violet. That stains purple. This one stains purple. So because of its fake peptidoglycan layer, is able to absorb the crystal violet more, and hence does not give room for the decolorization to occur. But for the gram-negative bacteria, so gram-negative, for the gram-negatives, so for them they have thin peptidoglycan layer. So they have thin peptidoglycan layer. So what do you think will occur? So during a gram stain, when it's reached the decolorization process, due to its thin peptidoglycan layer, all the crystal violet components of the stain will be washed off, or will be decolorized. Then upon the application of the counter stain, it will be able to stain the component pink. So to stain the component of the cell wall pink. So this is how we are going to know that this particular cell was what is a gram-negative bacteria. Okay, so this is a process that is done called gram staining. All right, so that will be for the cell walls. Now, in the cell appendages, we also have the cell membrane or the plasma membrane. So just like the plasma membrane of other cells, if you haven't watched our plasma membrane video, you have to go and watch. That same phenomenon occurs, selective barrier that controls the movement of substances in and out of the cell. So the cell membrane of the prokaryotic cell is just like the cell membrane of the eukaryotic cell. So if you haven't watched our videos on structure of the plasma membrane, you have to go and watch. Alright, so now let's focus our attention on this one, the cellular appendages. So the cellular appendages, we have the flagellum, we have the pili, we have the capsule, and we have the fimbriae. Now let's talk about the flagellum. Now, flagella. So the flagella is basically the thread-like substance that is found within. So let's draw this one to be a beautiful flagella. Okay, so the flagella is actually a thread-like substance that is found in the bacteria, and it enables the bacteria to move. So it's basically for movement. So now the flagella has basic parts. So first of all, we have the filament, we have the hook, then we have the basal body. So this filament is the longest and most obvious. And the hook is short and curved. So the hook actually serves as a link between the basal body and then the filament. And the basal body too is a complex structure. The basal body itself is a complex structure. So the basal body is embedded within the cell. So if you look at the structure of the bacteria, you realize that the basal body is found within the cell. So the basal body is embedded in the cell, and it has its own complex structure. So they are actually made up of rings. So for the gram positive, their basal body have 2 rings. And then for the gram negative, their basal body consists of 4 rings. So this is all you have to know for now about the structure of the basal body. Alright, so we have to know that the flagellum has 3 parts. The flagellum is firstly responsible for the movement of the bacteria, to propel the bacteria forward. So we have 3 parts: the basal body, the hook, and then the filament. Okay. Now the flagellum has its arrangement and how specific those arrangements have been formed. So first of all, we have monotrichous, then we have amphitrichous, we have lophotrichous, then we have peritrichous. So mono means 1. So for bacteria that have a single polar flagellum, it's called monotrichous. So for instance, if this is a bacteria Bacteria has a single monoflagellum. So this is a monotrichous bacteria. Then we have the amphitrichous. So one flagella is here, another flagella is here. So that is amphitrichous. And then lophotrichous. That one we have tuft of flagella at one pole. Then the peritrichous, they have flagella surrounding it. So we have some here, we have some here, we have some here, we have some here. So that is how bacteria too are classified based on their flagellum.